I want to talk about reverse translation and what I mean by translation, not just in the traditional sense from bench to bedside, but in other circumstances. And more importantly, I want to talk about or explain why it is I do large clinical trials and how that might relate to other types of research. So the more traditional translational pathway, and one highlighted here in the Cooksey report from the UK back in 2006, where they were trying to, I guess, improve the efficiency and focus of research in the UK and looking at the different research agencies that were working at that time. Now, I highlight this because I believe personally that this is the way it's moving in Australia, albeit relatively slowly. And the more typical translation pathway from bench to bedside and from clinical studies to clinical practice are often described as the big gaps in translation, and that is no doubt true. But what I want to really focus on in this presentation is really to think about, well, how can we actually take the clinical research activity that's going on, in my case, clinical trials or the clinic or hospitals, and actually take advantage of that activity and funding to open up opportunities and improve mechanistic insights that lead to how things do or do not work, or in what groups, why they work in some groups of patients and not others. And this concept of embedding research into practice is actually the way that it's been moving in the clinical research world in the last 5 or 10 years, to actually have learning hospitals, that every patient coming into the hospital is enrolled in at least one clinical study so that the clinicians and those that support them can learn from that experience and the results of studies, and then obviously more quickly integrate advances in knowledge into improved care. And the presentation I'm giving is my personal bent. All I really do is large clinical trials. I guess that's where I get my support from NHMRC and elsewhere. And we're currently conducting 5 large randomised trials right now, all funded by the NHMRC to a total of amount, certainly in excess of $20 million. And those top 5 studies, 4 of those were funded by NHMRC. So what is it about randomised trials? Now, I missed the talk before morning tea, but I think some of these aspects were highlighted there. But David Sackett, who is an epidemiologist and was the father of evidence-based medicine, he's a Canadian, spent a number of years in Oxford, sadly died last year. And he made the point that the intervention trial of greatest benefit to patients satisfies 3 objectives. So as a doctor, I'm very interested in benefit to patients. My whole focus is improving the experience of patients having a surgery and recovering well, having a good life. All my research is premised around those aspects, and my grants are written to highlight that feature. It's not necessarily just to do a study because it'll make me famous or whatever else. So coming back to his 3 objectives, validity, its results are true. Very important. Generalisability, the results are widely accepted and can be used in everyday practice around the world. And lastly, efficiency, actually saving resources for better patient care, not wasteful treatments that don't work or are so expensive and only have a trivial benefit over a much cheaper or safer drug. And he makes the point, of course, the first objective is so crucially important because if you get that wrong, you're heading down the wrong roads. And way back in the early 1990s, when I was trying to sort of build up my own research career, I was trying to see how can we, anaesthetists, do better quality clinical trials. And at the time, the average size of a clinical trial in the anaesthesia or surgery world was to enrol 60 to 80 patients and come up with some version of truth. It was patently ridiculous. And more importantly, the sort of endpoints we were studying were sort of trivial or surrogate endpoints of real no direct connection to patient outcomes after surgery. And the problem is Serious problems after surgery like stroke and heart attack and death are very rare. Modern surgery and anaesthesia are quite safe. And you need to literally study thousands, if not tens of thousands of patients to have a valid trial, to have enough statistical power and certainty to find a true effect. The way we overcame this dilemma was not just to enrol much larger sample sizes in our studies, but to focus our efforts on higher risk patients who are much more likely to have serious complications. So the event rate is much higher, you get more bang for your buck, if you like, and focus on the groups that are most likely to benefit. And this concept's been extended a little further about this idea of practical clinical trials. In other words, we study real-world settings, everyday patients with treatments that anyone can do almost anywhere in the world, teaching hospitals, small rural hospitals, hospitals in South Africa, New Zealand, Australia, the US, and so on. And we actually look at meaningful outcomes for patients. And those ingredients get built into the trial design so that when the trial is done, the results are immediately translatable. Not just because the results are meaningful, but the people doing those studies have been working together across a network so they know of the clinical problem, the uncertainty. When the result comes through, they can enact it in their clinical practice literally weeks later. So there isn't this T2 or translation gap needing years before people finally hear about effective or ineffective treatments. Now, one example I'd like to talk about is our ITACS trial. Each trial I'm going to talk about today has got an acronym, just the things you do to get funded. ITACS is aspirin and tranexamic acid in coronary artery surgery, and one of the main aims of this was to focus on tranexamic acid, or TXA, because there was a concern that this might increase thrombotic complications after coronary artery surgery. It was a big international factorial trial looking at both aspirin and tranexamic acid. We enrolled 4,600 patients having coronary artery surgery, and our primary endpoint was 30-day death or serious thrombotic complications, myocardial infarction, stroke, etc. We had typical range of secondary endpoints, which are other serious complications after surgery. And we published both of these papers over a 12-month period in the New England Journal of Medicine. Great for me, but also giving high impact to the results and translating into everyday practice much quicker. Now, for those of you that don't know, tranexamic acid is known as a lysine analogue drug. It actually binds to the lysine receptors on fibrin to block the action of plasmin, which breaks down blood clot, fibrinolysis. It's an anti-fibrinolytic drug. And the reason why we believe this might increase thrombotic risk, and this is crucially important for people having coronary artery surgery, is because there was a whole lot of case reports, uncontrolled studies, retrospective studies, mechanistic basic science studies, all suggesting that in fact you can get extra thrombosis or extra clotting problems if you block the body's innate fibrinolytic pathways. And there was enough concern and doubt for us to do the study. And this is just part of the results of that study where we clearly demonstrated the beneficial effects of this drug in reducing bleeding, reducing the need for blood transfusion, the amount of blood transfusion, and most importantly for patients, reducing the need for them to return back to theatre for emergency surgery because of life-threatening serious hemorrhage or cardiac tamponade, which most commonly occurs after midnight after the first day of surgery. Incredibly high risk. Patients who go back for further surgery have a 10-fold increased risk of death. Not to mention the cost of doing that, the inconvenience of dragging a lovely anaesthetist back from home in the middle of the night to do this really life-threatening surgery. A pain in the neck, but also both expensive and all the rest. Now, I'm glad Rob Metcalf's in the audience here because his PhD student and himself have been working in this area for many, many years. And we've in fact linked up because of my interest in tranexamic acid and doing this clinical trial to actually look more closely at some mechanisms that might explain both the results of tranexamic acid in the cardiac setting, but also the trauma setting, and into the future looking at other types of surgery. And Rob taught me that, in fact, plasmin has more effects in the body beyond it being a fibrolytic part of the fibrolytic activity. And we've already been collecting samples from the ITACS trial and now our other studies to actually go back to the lab and look at some other mechanisms that might explain some of the clinical effects of this drug. So a reverse translation effect, but equally coming up with new hypotheses or new ideas, proper new discovery research to then go back to the clinic setting and for me to design the right studies to test those future hypotheses. And at the same time, in fact, we've got a whole suite of genomic studies being led by Chris Bain, who's an anaesthetist doing his PhD right now in this area. So he's got a background in science himself. He did honours science in genetics before doing medicine and now anaesthesia. He's a fantastic, high-quality clinician anaesthetist, but he loves basic science. And he's building up a whole suite of studies from our clinical studies to collect samples not just at baseline, but also through the surgical pathway, looking at epigenetics and potentially into other areas here with the labs here. Now, the next big trial, though, the one that we've just finished, the RELIEF trial, looking at restrictive versus liberal fluids in major abdominal surgery. It's a very simple premise. The traditional practice for anaesthetists for more than 50 years is to set up an IV and give patients intravenous fluid. They're fasted for surgery, they often can't eat, they lose blood, they get dehydrated. So we give them fluid maintenance and resuscitation. And like people with vitamins, some people think IV fluid is so good that you should give lots and lots of it. And during surgery and soon after surgery, it is very common for patients to be hypotensive, to have low blood pressure, possibly low cardiac output. So the anaesthetist or the other doctors and nurses looking after the patient give them fluid boluses, lots of fluid intravenously. And if they get more unstable, they get more and more fluids. And there are patients at the Alfred Hospital that get 10 or 20 litres of crystalloid fluid during and the first 12 hours after surgery. I can tell you, if you step back from that everyday clinical practice, it is patently ludicrous that you can actually make someone well giving them 20 litres of salty fluid. The body, which has developed, I think, over millions and millions of years, evolved to actually protect itself against starvation and dehydration. The kidney is an immensely complicated organ that can help defend the body against dehydration or shock. The body has never evolved to deal with getting 20 litres of fluid in hours. We literally drown our patients in intravenous fluid and make the tissues very boggy or edematous and probably cause much more harm than good. So, in the last 5 years or so around the world, there's been this move towards restrictive IV fluids in many types of surgery, in critical care and in other settings. So the dilemma becomes for me, well, what's better, a more restrictive approach or a more liberal approach? And that's what we put up to NHMRC. They accepted this was a clinically important question. They funded the study. We've finished enrolment and we're doing all the analyses right now. Our primary endpoint is disability-free survival out to a year after surgery, and we're looking at serious complications in the weeks and months after surgery as well. Now, built into this single large trial, which has got millions of dollars attached to it from the NHMRC and the New Zealand Health Research Council and the UK National Institute of Health Research, they've all put lots of money into this study. We've built in a whole lot of mechanistic studies to look a more focused, smaller sub-study around kidney injury or sepsis, or wound healing, or nutrition, or the relationship of obesity and complications after surgery, and how that might interact either with fluids or these other complications. And in fact, 4 of these studies have been funded by the Australian New Zealand College of Anaesthetists just for the mechanistic basic science linked sub-study, typically in 50 or 100 patients. So that's pumped in another $500,000. Others that aren't yet got the extra funding have got PhD students attached to them, both medical and non-medical PhDs. And this is one example of that. This is Chris Bain's genomic sub-study, which he's had funded through the college. And he's looking particularly around the effect of inflammation. Why is it that patients get inflamed? We know why, it's because of tissue injury, but how can that be mitigated? At what point is that good or actually destructive for wound healing? And he's doing a lot of work around that area. Now, the third trial I want to briefly mention is our ITACS trial. This is our most recently funded large trial for NHMRC. So we're looking at an intravenous iron injection in the weeks before elective cardiac surgery. It's such a simple, cheap intervention, something that couldn't really exist 10 years ago because of the safety of that medication, but there's now much cheaper products. It's an international trial once again. We're taking 1,000 patients having elective cardiac surgery. We're measuring very typical clinically focused endpoints in the study. Now, why are we doing this study? Well, there's lots and lots of epidemiological data, registry data, clinical studies that show that patients who are anemic having cardiac surgery, or in fact any major surgery, have a higher risk of complications, higher risk of death, longer stay in hospital. They also have a greater need for blood transfusion during and after surgery. And both anemia and blood transfusion are both independently associated with much poorer outcomes. And we know that if we treat anemia, we can reduce the need for a blood transfusion. That much is already known. So that alone might be good enough reason to use intravenous iron. However, there is some basic science suggesting that intravenous iron as an injection might lead to increased infection risk. Bacteria quite like iron, like tissue cells in the human body. They get energised by iron and they therefore might increase infection risk. So we've got a cost, an inconvenience, and perhaps a theoretical risk by using this in the clinical setting. And as I mentioned a moment ago, the contemporary or modern iron preparations are far safer than the older products. They're licensed for use in Australia, and we can give a single dose of intravenous iron over really 5 or 10 minutes. It's the equivalent of 1 year's tablets to treat anaemia. So it's effectively a sudden, incredibly effective treatment. But again, we don't know whether that actually improves outcomes after surgery, whether we should delay surgery until they get a hemoglobin response or whatever, and so forth. What we do know from a group from the UK that did this randomised trial and published in the New England Journal a number of years ago is that patients with heart failure in the non-surgical or community setting, that if you treat anaemia with a single injection or a course, I think, of low-dose IV iron, that they have much improved symptom and function, that they actually have better quality of life purely from that very simple treatment. And many patients having cardiac surgery, in fact, do have heart failure, so that might be the reason. And what we're wanting to look at now is, well, what are the underlying mechanisms? Because the problem with patients having the surgery, because of chronic disease and heart failure, they have this low-grade inflammatory state that might be the explanation of why they have anemia, this so-called functional iron deficiency or anemia of chronic disease, related in part to upregulation of hepcidin, a compound that actually, released by the liver, that actually effectively controls iron uptake in the gastrointestinal system. Now, we don't know that IV iron can be effective or is effective in this surgical setting, or what types of patients does it work in or not work in. So Joel Simons, who's again, he's an anaesthetist in our department, he's doing his PhD around this underlying mechanisms of what level are patients actually going to be responsive to the iron therapy or non-responsive. So that means in the future we can target the right patients in a preoperative anaemia clinic, which would fundamentally change how we manage surgical patients here and around the world. So that's really the focus of his talk. I'll skip through that. So he's actually looking particularly at other markers that might inform us about why patients would or would not be responsive to IV iron therapy, and in particular taking blood samples before, during and after surgery, and bone marrow directly from the sternum during sternotomy to actually inform his thesis. So I'll conclude here with that same slide I showed you earlier on. We often talk about and think about this translation pathway, and I just want to remind you it's not a one-way process. I actually start nearly all my research with guidelines or practice as it exists, and I ask the question, why are we doing that? What's the validity of the underlying science that would inform that to be actually the best thing for patient care? And so often there is so much uncertainty and conflict in the literature that it allows me to mount an argument to do a clinical trial. And I'm now wanting to work more closely with particularly basic scientists to go further back in that reverse translation step to try and understand those reasons. Thanks very much.